Hi everyone, I am Joe Poblete, and in behalf of my colleagues from the University of Maine, our, I am presenting our study entitled Efficacy of Binders on Per and Polyfluoroalkyl Substances, or PFAS, and Aflatoxin B1 levels under in vitro ruminal conditions. So for the introduction, I'm pretty sure most of you have heard or known about PFAS. So they are man-made chemicals that are found in a wide range of products. They include firefighting foams, non-stick cookwares, food packaging, pesticides, and among others. They are chemically stable and heat resistant, and they can easily repel oil and water. So these very same characteristics are the reasons why they are very difficult to be degraded in the environment. Thus, they are oftenly referred to as forever chemicals. So it is often attributed to their chemical structure. As you can see, there are a lot of fluorine atoms that are attached to their carbon chain. So these carbon fluorine bonds are among the strongest bond known in chemistry. So perfluorooctane sulfonic acid, or PFOS, is a type of PFAS that has the highest tendency to bioaccumulate in animal and human tissues and cause serious toxic health effects. Then what's the reason why PFOS contaminates beef and milk most, and it's very difficult to be excreted in the body? So it has something to do with the enterohepatic circulation. Based on our current understanding of PFOS, it hijacks the enterohepatic circulation and uses it to stay inside the body. It is reported that PFOS mimics the action of bile salts and use similar transporters such as OATPS, NTCP, and ABST to move across the hepatocyte and or enterocytes. So it is reported that a huge amount, around 42% of PFOS in the ileum part of the small intestine gets reabsorbed through the enterohepatic circulation. So as you can see, a small fraction of PFOS are eliminated via feces, and only 1 to 14% of PFOS are eliminated via milk. Furthermore, the presence of sulfonate group and long carbon chain of PFOS makes it difficult for the kidney to filter it out and excrete it out of the system. By the way, do you know that Maine is among the few states that has established action level for PFOS in milk and in beef? So in milk, it is 210 parts per trillion, and in beef is 3,400 parts per trillion. Aflatoxin B1. So it is a carcinogenic mycotoxin produced by Aspergillus flavus and Aspergillus parasiticus. They are transferable from contaminated feed to the milk, so they get metabolized into aflatoxin M1 in the liver of the animals and can be excreted to the milk. So once a human ingests a large amount of milk contaminated with aflatoxin M1, it can result to reduced growth and development, immune system, higher risk of birth defects, and hepatocellular carcinoma. So as you can see in the title, so we are using binders for this experiment. So what are binders? Binders encompass virus sequestering agents such as bacteriocels, yeast, protein, and clays. So one very good example of that are the aluminosilicate clays. These aluminosilicate clays have a negative charge in their surface, thus they can able to attract positive charges from unwanted chemicals and toxin. Furthermore, these clays have a porous structure and has a very large surface area that is responsible for its ability to bind large amount of chemicals and toxins from the feed. So let's go on to our objective. So the objective of the study is to evaluate the efficacy of binders in sequestering PFAS and aflatoxin B1 under in vitro ruminal fluid conditions. So let's go on to the materials and methods. For aflatoxin B1 analysis, this serves as the positive control for binding. So we just want to test out that the assay that we will be using is effective in testing out the effectivity of binders for PFOS under in vitro ruminal conditions. So for this aflatoxin B1 analysis, We use alfalfa as substrate, and then for the treatments are control, and then the clay binder 1 and 2, which are clay minerals that have very good cation exchange capacity. So we use a randomized complete block design with a total of 3 blocks. For the in vitro ruminal incubation, we did this in sterile conditions because aflatoxin B1 can easily be degraded under ruminal conditions. So we use 0.05 gram of binder, and we add it to 3 grams of ground substrate plus 75 ml McDougall buffer plus 25 ml of ruminal fluid in a 250 ml bottle. And we added aflatoxin B1 from Aspergillus flavus, 10 parts per billion concentration in the media at 0 hour. Right after that, we incubated for 2 hours at 39 degrees Celsius and 60 revolutions per minute. Afterwards, the contents were centrifuged, and we collect the supernatant and analyze it for its aflatoxin B1 concentration using liquid chromatography mass spectrometry. Now let's go on to the PFOS analysis. For the substrate, we use Timothy grassy that is collected from a PFAS contaminated site abandoned in 2020. And we also use a randomized complete block design with a total of 4 blocks. For the treatments, we have a control group, a clay binder 1 and 2, similar to the ones that we use in the aflatoxin analysis. And we also have a polysaccharide binder, a natural biopolymer used to bind mycotoxins, and a carbonaceous binder, a binder with high surface area and a porous structure. And we also use anion exchange resins 1 and 2. These are binders which can remove anions from aqueous solutions. Now let's go on to the in vitro ruminal incubation. So we use a 0.05 gram binder, except for the anion exchange resin 1, wherein we use only 0.01 gram. And we add it to 3 gram of substrate, 75 ml of McDougall buffer, 25 ml of ruminal fluid in 250 ml bottle. And then we monitor the cumulative gas pressure using the ANCOM RF gas production system. And then we incubated it for 48 hours at 39 degrees Celsius and 60 revolutions per minute. Afterwards, contents were centrifuged, and we collect the supernatant and analyze it for its PFAS concentration using the APA method 537.1. So right after collecting the data, we proceed to the data analysis. We use the SAS-proc-NLIN procedure to estimate the asymptotic maximum and rate of gas production in PFAS bottles. And then we calculated the aflatoxin B1 and PFOS binding affinity in relation to the control using this formula. We subtract their concentration in the treated rumen fluid from their concentration in the control rumen fluid, and then we divided it by the PFOS or the aflatoxin B1 concentration in the control rumen fluid, and then multiply it by 100. Right after that, we use the proc-Limix to analyze the effect of binders in the binding affinity, and then the gas production kinetics of the PFOS bottles. Means were separated by a pressure-protected LSD. So right after the data analysis, let's proceed to the results and discussion. This table shows the aflatoxin B1 binding affinity with binders under sterile in vitro ruminal condition. So the y-axis indicates the percent aflatoxin B1 binding affinity. So the block bar represents the clay binder 1 or the bentonite, and the gray one represents the clay binder 2, which is the aluminum pillared montmorillonite clay. So as you can see, around 80% of aflatoxin B1 bonds to the clay binder 1, so which is 40% greater than the amount that bound to the clay binder 2. So this is statistically significantly higher. And then this also confirmed that the assay that we use validate the effectivity of this procedure in testing out the binding effect of different binders for PFOS. So let's go on with the PFOS binding affinity. need binders under standard in vitro ruminal conditions. And we did this in a non-sterile conventional uh, condition. And similarly, the y-axis represents the percent binding of PFOS, and then the black bars represents the clay binder 1, the gray bar is the clay binder 2, and then the green bar is the polysaccharide binder, the blue one is the carbonaceous binder, the yellow one is the anion exchange resin 1, the red one is the anion exchange resin 2. So as you can see, it is very noticeable that an anion exchange resin 2 bound more than 50% of PFOS relative to the control. So this anion exchange resin 2 is the only binder in this group that has the presence of quaternary ammonium group. This quaternary ammonium has a positive charge which attracts the negatively charged conjugate base of PFOS under aqueous media. Furthermore, the PFOS and anion exchange resin 2 have a both hydrophobic nature, which means they tend to attract each other and form a compound that is large enough that it cannot be absorbed by the body, and it tends to be excreted out of the system. So for this table, the gas production kinetics of PFAS contaminated Timothy grass with different binders under in vitro rumen conditions. As you can see, the p-value is more than 0.05, so there's no significant differences in the rumen fermentation kinetics. Thus, it confirms that anion exchange resin 2 can be safely fed to the animals to bind the PFOS without negatively impacting the rumen fermentation. So for the conclusion, so under in vitro ruminal conditions, clay binder 1 exhibited a high ability to absorb aflatoxin B1. And this confirms the validity of the assay that we use in this study. And then anion exchange resin 2 demonstrated the best potential to sequester PFOS. And then for the future research, we are looking to explore the use of anion exchange resin 2 and other similar binders to reduce the PFOS contamination in vitro. And then the binder that shows the best potential will be tested out to possibly reduce PFOS contamination in milk under field conditions. And then here are the list of references that we use for this study. And then for the acknowledgement, we would like to acknowledge the funding that was provided by the Maine Food and Agricultural Center, the Maine Department of Agriculture, Conservation and Forestry, and the American Farmland Trust. And that's it. Thank you so much for listening and have a good day. Bye.